So hello everyone, and a very warm welcome to this exclusive Genflow Bioscience interview. My name is Colin, so thank you all for joining us via YouTube or podcast. Today our special guest is Dr. Vera Gorbunova. She is a professor of biology at the University of Rochester in New York State, and she's also co-director of the Rochester Aging Research Center, where her research focuses on the mechanisms of longevity and genome stability. And for those not familiar with Genflow Bioscience, Genflow is Europe's first listed longevity company and focuses on developing gene therapies that target the aging process. And it also has some very exciting developments in the treatment against NASH, a liver disease that affects millions of people worldwide. So we'll be talking about both of those later. But good morning, Vera.
Good morning.
It's very nice to see you again. How are you keeping?
Very good, thank you.
You said you had a bit of a cold when we just talked just now. Just a sore throat or a sniffy nose?
Yes, well, kind of a virus. Traveled to several conferences in Europe, and yeah, that's where you catch viruses.
Well, look, I guess you know about viruses, but firstly, I would like to congratulate you on your recent promotion to the chair of Genflow Scientific Advisory Board.
Thank you very much.
Was that a bit of a shock when Eric asked you to take on this role?
Well, it made sense because of, how involved I am in SIRT2 and 6 research. So, it wasn't a shock.
Yeah, I know he was very excited. It was great to read that in the RNS. But look, well, congratulations again. So look, just to get everybody up to speed, for any newcomers that are listening, we'll start with some background on Genflow's initial research on SIRT2 and 6. Can you give us a quick recap on what SIRT6 or SIRT2 is and how it relates to aging and longevity, please?
Yes, SIRT2 is a gene, a very interesting one, because we can definitely call it a longevity gene. And what is a longevity gene? So perhaps a gene whose function is to contribute to longevity, to ensure healthy lifespan of an organism. And how do we know it? If you put SIRT2 in a mouse, like give it an extra copy of this gene, mice live longer. They can live up to 20% longer. And if you remove this gene from a mouse, then they become very progeric. They have a lot of problems and that resemble aging, and they age very rapidly and die within a few weeks. So that's what makes SIRT2 a longevity gene. Obviously, after we learned about these properties, so then we want to find a way to boost it, because the more activity you have, it's supposedly better for your health. And maybe another couple of words of what it's doing, what makes it help longevity. It makes a protein. This protein is found in the nucleus of the cells. And there in the nucleus, it takes care of the cell's DNA. Because DNA, this is the molecule that encodes genetic information, that gives instructions for the cell how to live, pretty much. But it needs a lot of care because it's a long molecule, it can get damaged, needs to be repaired. Also, because it's such a long molecule, like a very long text, it needs to be packaged very carefully. So every time the cell needs to access particular gene in the DNA, it could find it. And that is really a problem, because if you think how long this strand is. So what SIRT2 does, it has a role in repairing DNA. So if it gets damaged, it comes there and it helps orchestrate repair. And it also helps package DNA in a neat way so that cell can find what it needs when necessary. And with aging, both of these processes start to deteriorate. We get more damage to DNA, and those strands of DNA get messed up. So that's where we can benefit from more SIRT2 because it can come in and repair DNA and also arrange it in a more youthful and neat way.
Now, I think one of the most interesting findings about SIRT6 or SIRT2 is the discovery of the centenarian variant of this gene. So look, can you just tell us how this version of the SIRT6 differs from regular SIRT6? And what are the implications of these differences for aging and longevity? Is there much of a difference between the two? I mean, centenarian would indicate somebody lives 100 or more years of age.
Yes. No, this is super exciting because, so far I was just saying, well, more SIRT6, more of the same, but can you also make whatever you have work better, right? And there are some very fortunate individuals that lived up to age of 100 and beyond, centenarians. Many of them are very healthy, so not only they lived up to 100, but they also stay active into their 90s, and they continue to work, enjoy life. So that's something all of us would like to be able to do. And what we found by collaborating with people who study human genetics and study cohorts of centenarians, and when we looked into SIRT2 and 6 and centenarians, we found a variant that was more frequently found among centenarians. It's very rare, so it's not to say every centenarian has it. So it's very rare, but in sort of randomly taken individual, you would probably have zero chance of finding it. But in centenarians, it can get, you would have a higher chance of finding.
Is there a certain part of the world where there are more? I mean, I know Japan has lots of people that live over 100. I think in some places in Italy as well. Does that centenarian gene imply that it's more in Japan, or it's in certain parts of the world, or is it all scattered everywhere?
So the original finding we made in the cohort of Jewish centenarians that live around New York area, and that's primarily for historic reasons because we had access to DNA from this cohort. Then we don't really know about other ethnicities. And that is something then because that was a question, okay, we found it in Ashkenazi Jewish centenarians, can we find it anywhere else? And to be able to replicate this, we would need access to a large number of centenarians of different ethnicities. And this is often a limiting factor because, human genome sequencing is becoming affordable, but you not only need to sequence enough people, but you need to sequence enough centenarians. And then we actually tested on a larger cohort of all mixed ethnicities, just whoever happened to be in the cohort. And there were thousands of people, tens of thousands, very mixed background. And there were not very few centenarians in that cohort because you need to specifically look, these people are very rare. But what we were able to do is just to look at people young, different ages, young people and people who are older than, I think we put a cutoff at 70 years old, which would mean that if we have these people, they made it to 70. So that means they probably had some health advantage which allowed them to make it to this age. And when we just divided people by, everybody young and everybody 70 plus, we saw this allele to be more frequent in older people.
Okay, I was recently came across an article mentioning there are wild versions of genes. Of course, I assume this applies to SIRT6 too. What is a wild type of SIRT gene, and what are the benefits or advantages of centenarian SIRT6 when compared to this wild type?
So wild type, it is a little bit funny when this term is applied to humans, because mostly that's how people talk about animals, mice, for example, because, you could have a wild mouse, and then that would be like your normal genotype, wild type, and then something that is bred in the laboratory would be the opposite. So when you talk about humans, it's usually more of a,
well, I know plenty of wild humans.
Yeah, because all of us are wild type.
Of course.
And so wild type human would be, just a human. Wild type SIRT6, there is a canonical sequence that you can find in a human gene bank database. There are some, we are not all identical. So there are few, alleles that don't seem to differ from each other very much. And I think there are probably like only maybe two major variants of SIRT6 that seem to be very similar functionally. And then there is this centenarian variant that, of course, you don't see in sort of wild type people or in sort of wild type database. It has two amino acid change in the C-terminus of the protein. C-terminus, it's like the tail of the protein. There are two amino acid change, so it's still a functional protein. It makes a full-length product, but there are two amino acids are different, and it makes SIRT6 better at those two functions that we discussed. It makes it better at repairing DNA, and it is better at packaging the threads of DNA into the right conformation.
I mean, it's so in layman's terms, then the SIRT6 gene, how does it influence the aging process and cellular health? Does it like is this natural? Can you add this to the gene and change things around, or is it just born with what you're given with?
Well, we are all born with two copies of every gene. But this is like genetic material, that's the DNA. But then, of course, you make a protein based on this, on your DNA, and the levels may differ slightly because this is a very complicated process. There are multiple factors that can affect how much protein is produced. So there may be some individual differences between people. And from what we see, that at least from the animal studies, that if you make more of SIRT6, that seems to bring better health. And then in addition to it, you could have a variant in the sequence, like what we discussed, centenarians, that may produce enhanced protein, but improved function.
Okay, I mean, I also see recently that Genflow announced that they've made some remarkable strides in their preclinical program, which is aimed at addressing liver fat deposits, fibrosis, and liver cancer, all of which seem closely linked to NASH. Now, I know it's a slightly different subject, but I think they were linked. So I assume that the successful outcome of treatments holds the potential to profoundly impact the lives of millions of people with NASH. So that begs the question then, from your perspective as a scientist, of course, what actually is NASH and how does it negatively affect the liver?
Yeah, so NASH is a disease of aging in some way. Well, that's with age, people would lose function of their liver. There are multiple sort of predisposing factors. It could be related to alcohol consumption. It could be related just to the process of aging and the metabolic syndrome as well. But all those multiple factors lead to replacement of healthy liver cells with just fibrous tissue and fibroblasts that cannot function anymore properly. And this is a very difficult condition to treat. There is no cure for it because you can really look at it as aging of the liver tissue. It just becomes old, it loses capacity to replace, like cells die, and they can no longer be replaced with functional liver cells. Instead, there is just some connective tissue that replaces it, and liver function is lost, and without functional liver, people cannot live. So this is very serious because liver is very important to clean our blood and detoxify. So liver function is really vital. And that is a very big health problem because once people start losing function of their liver because of fibrosis and NASH, there is really no way to reverse it. And here comes this very hopeful idea that if we apply SIRT6 to the liver, we give liver extra dose of SIRT6, it may be able to reverse NASH by rejuvenating the liver, because if liver fibrosis is so difficult to treat because it's a reversible aging process of the tissue, then maybe, let's bring SIRT6 to the rescue, and if it can rejuvenate the cells in the liver, they may be able then to repair the organ and produce functional, youthful cells again and fill those gaps in the tissue.
So what are the mechanisms of action then for the SIRT6 gene, and how is that modulated in the liver? How does that actually work?
So the function would be the same that we just discussed. So SIRT6 functions in the nucleus. It helps repair DNA, and it helps repair packaging of the DNA. Because with aging, what happens, the arrangement of DNA becomes disrupted. So the cell now can no longer properly activate genes when necessary. It cannot shut down certain genes because our DNA is full of sort of parasitic sequences. They're called transposable elements or repeated sequences. And usually in the young cell, those repeats are silenced, so they don't disrupt anything. But with age, they start to rise, and they promote inflammation. And that is also the mechanism, one of the mechanisms that drives aging, that drives fibrosis of tissues, because there is more inflammation. So what SIRT6 would do, it come in and shut them down again, like in a young cell, and also organize the rest of the chromatin in a way that it's more like in the young cell, so it can now function properly. It will give rise to more youthful cells that can then repair the organ. So that is what we expect SIRT6 to do in this model, because there are kind of conceptually two approaches for treatment of such diseases. Either, you try to treat the symptoms and indications. And in the case of diseases that are actually just manifestation of the aging process itself, which is the case with fibrosis, this is not very effective because, you can do something temporarily, but it really doesn't reverse the process. Or you can try to just come and say, we'll try to make the tissue younger, and then it will repair itself, because liver has a great capacity to repair itself in a young person. It can, people would donate, part of the liver, and it just grows back if you are young. But when fibrosis hits, then it can no longer repair itself. So there is this capacity in young liver. We just need to try to bring it back.
So are you quite excited about the potential for SIRT6 for treatments of NASH?
Yes, I'm very excited.
Me too. Look, I think the liver is quite remarkable. It's, as you said before, its regenerative capacity is sort of widely acknowledged. I mean, I can just imagine the potential of if SIRT6 could be demonstrated to effectively combat liver diseases such as NASH. I think it's an incredibly exciting prospect. But with that then, are there any exciting or any specific ongoing studies or clinical trials, apart from what you're doing, that are investigating the effects of centenarian SIRT6, or of course its wild brother, in patients with NASH?
So one direction is NASH, because liver is the organ that is more easily accessible with gene therapy, and that was our first focus, and also because of the importance and in the society of how widespread this is so-called unmet need, because there are so many people affected by it and there is no good cure. But of course, ultimately what we would like to pursue, and that's something we are doing right now in my laboratory, is to just try to address aging altogether. Because, there are diseases of aging like NASH, and then there is aging that may not be associated with, particular disease, but people just get frail, they lose their capacities. And we want to see whether SIRT6 can alleviate that and reverse this process.
I mean, it's obviously still in its early stages, but really has the potential to revolutionize the field of medicine, I think. And not only in treating NASH, but probably also combating cancer, metabolic disorders, and maybe even neurodegenerative diseases.
Oh, yes, absolutely. There is a link between SIRT6 and Alzheimer's disease. Yes, there were studies published showing that people with Alzheimer's have lower level of SIRT6 in the brain. So, maybe gene therapy for the brain would be a little bit more challenging than liver, but it's still something to consider, and there could also be pharmacological ways to boost SIRT6, and it may help neurodegenerative diseases as well.
So, with it then, do you see other potential applications or areas of research that are being explored for SIRT6? And if that's the case, how might these discoveries contribute to our understanding of aging? I guess the development of future treatments.
Well, so the way I see it, this approach we are taking now with the liver fibrosis could be applied to other organs as well, because lung fibrosis is a very big problem. So it can be, if we see success with liver, we can also look at lung. We can also look at various cancers. I think the problem with SIRT6 is because there is this systemic improvement, at least that we see in preclinical models. We just need to focus, because it seems we can apply it to multiple diseases of old age. We just need to prioritize which one we address first.
And how long do you think we'll start seeing human trials? Will that be in a couple of years' time, or are they going to start with dogs and larger animals first?
Well, if we see very promising results right now, I would think human trials maybe within 5 years. I think it depends really of what we see, because there is a huge need.
Yeah.
And I think that will not take too long.
Excellent. Well, that's great news. Well, look, I know you're very busy. I think that's an excellent ending to our webcast today, because I know you're in constant demand. So if you don't mind, we'll conclude the interview here.
Okay, well, my pleasure.
Thank you. So look, for those that would like to see more information, you can visit Genflow Biosciences website at www.genflowbio.com, or you can email them at info@genflowbio.com. So Vera, thank you once again for your time. I wish you all the best in your new role as chair of the SAB, and I really hope you have a wonderful day. My name is Colin, so thank you all for joining us via YouTube or podcast. Thank you. Take care. Bye-bye.